Okay, so before we get into this antibodies, the structure, we're just going to highlight some points from the previous lecture. Okay, so IL-4 increases the Th2 population. What is IL-4? So IL-4 tells B cells to switch to IgE, and IL-4 is also up in parasitic infections. Okay, so IL-4 increases Th2, right? What is Th2? Remember we talked about this? Um, CD4 T cell turns into helper T cell, and helper T cell becomes Th1, Th2, and Th17 and T regulatory cells. And IL-4 is up in Th2. So Th2 is what activates the eosinophils and increases IgE. So that makes sense, right? IL-4 tells B cells to switch to IgE. Um, so yeah. So IL-4 tells B cells to switch to IgE. So it tells B cells to make the IgE antibody. Okay. So which means that IL-4 increases Th2 population. Yeah. So helper T cell, um, there's helper T cell first, and that turns to Th2 with the help of IL-2 and IL-4. So IL-4 increases Th2 population, and then when there's more Th2, that in turn makes more IL-4, and this will now activate the eosinophils and increase IgE in the body. So the end goal is to increase IgE. Okay. So IL-4 increases Th2 population. So if IL-4 increases Th2 population, which is the offspring of CD4 T cells, then that means that IL-4 will decrease Th1 population. Yeah, see, this inhibits Th1 responses. And Th1 is still a subset of CD4 T cells, but CD4 T cells has four babies, right? Th1, Th2, Th17, and T regulatory cells. And of this specific one, we're talking about IL-4 and Th2. So IL-4 increases Th2 population. And if IL-4 increases Th2 population, that means IL-4 decreases Th1 population. Because one goes up, the other one must go down. Remember that chart? That chart is very important. I'll show you just one sec. Because we already went over it, but it's easier when you look at this chart. So when we're talking about Th2 right now, so remember how we said CD4 T cell turns into helper T cell, and then helper T cell has four babies: Th1, Th2, Th17, T regulatory cells. So we're talking about IL-4. See this IL-4? IL-4 will increase Th2. In in turn, Th2 will secrete IL-4, IL-5, IL-6, and that will increase IgE. So when this goes up due to IL-4, Th1 must go down, which means when Th2 is up, uh, macrophages and cytotoxic T cells will be down. Okay, so this suppresses interferon gamma and Th1 differentiation. And the other thing that you should know is IL-10 decreases Th1 population. IL-10 decreases Th1 population. IL-10 is anti-inflammatory. What is Th1? Th1 is a subset of CD4 T cells, right? What does Th1 do? Th1 is, remember, cell-mediated. This is the one that activates the macrophages and the cytotoxic T cells. Th2 is humoral immunity. And you have to remember, cell-mediated and humoral immunity are both subsets of CD4 T cells. Okay, so IL-10 is anti-inflammatory, and IL-10 decreases the Th1 population, which means that IL-10 will decrease the macrophages and the cytotoxic T cells in the body. So basically, IL-10 will increase Th2 population. And Th2 population is what we just said. It's the IgEs. So IL-10 will increase IgE in the body. Now this next part, listen carefully because it's confusing. IL-12 will increase the Th1 population. Why? IL-12 is secreted by macrophages. So IL-12 will increase the Th1, which is the cell-mediated pathway. So what happens here is macrophages come and secrete IL-12, and this IL-12 will go to the Th1 and make more Th1, and when more Th1 is made, the Th1 will secrete interferon gamma, which activates even more macrophages, who can secrete more IL-12, who can make more Th1 cells, and this is how macrophages activate more macrophages in the body. The macrophages make the Th1, or specifically IL-12, from the macrophages, make more Th1, which make more interferon gamma, which calls more macrophages to the site of attack. So that's what that is. And this IL-12 will increase the Th1 population. And interferon gamma produced by the Th1 will also increase the Th1 population. So interferon gamma and IL-12 work together to increase Th1 population. And if interferon gamma and the Leukin 12 both increase Th1. That means that interferon gamma and IL-12 will decrease the Th2 population. Remember how we talked about when Th1 goes up, Th2 goes down. When Th2 goes up, Th1 goes down. So I'll just say it quickly one more time. IL-4 increases Th2 population. IL-10 decreases Th1 population. IL-12 increases Th1 population. Interferon gamma decreases Th2 population. Okay, so we already talked about how the macrophages secrete IL-12, and the IL-12 goes to the Th1s. Th1 goes to Th1 to make more Th1s, and the Th1s secrete interferon gamma to call more macrophages to fight. This is exactly what happens in tuberculosis. Okay? Okay. If there is a interferon gamma receptor deficiency, then you treat with antimicrobacterial drugs. IL-12 receptor deficiency. So, interferon gamma receptor deficiency. If somebody has interferon gamma receptor deficiency, then you treat with antimicrobacterial drugs. If somebody has a interleukin 12 receptor deficiency, then that means that person can't call, that means they can't secrete IL-12, right? So that means their macrophages, well, they can't secrete IL-12. Okay, so they can't call macrophages. So if there's no IL-12, that means nobody can go to the Th1 to make more Th1, so that Th1 can secrete interferon gamma, so which means that the macrophages cannot be activated. Okay, so somebody has interleukin 12 receptor deficiency, they don't have Th1, then they also don't have interferon gamma, and they don't have macrophages to come to the place to attack, the site of infection. They don't have macrophages to come to the site of infection to attack. So people with this deficiency will get tuberculosis easily. And you cannot give the vaccine. You cannot give tuberculosis vaccine. These patients, you can give them interferon gamma to increase the macrophages. So you had to give somebody with the interleukin 12 receptor deficiency, you have to give them interferon gamma. And if you have interferon gamma, you can increase the macrophages. If you exogenously give them interferon gamma, you can give the you can increase the macrophages because macrophages respond to the interferon gamma. So I'd like to explain further on what this was. The IL-12 receptor deficiency. Okay, this is confusing, so you have to listen very carefully. The IL-12 receptor is actually on the T cells in the natural killer cells, not the macrophages. The macrophages secrete IL-12, right? Macrophages secrete IL-12, and then the IL-12 receptor is located on natural killer cells and the T cells. Okay, so in this body there is normal or increased IL-12 levels when it comes to interleukin-12 receptor deficiency, because there's no problem. There is no problem with the macrophages. The problem is with the receptor. The receptor is absent. So the problem is with the T cells. So if the T cells are, I mean, the receptor on the T cells are impaired, which this means that you can't bind like the interleukin 12. So the interleukin 12 cannot bind to the Th1. Remember, Th1 is a type of T cell. It is a subset of CD4 T cell. So I like to think of it as IL-12 that is secreted by the macrophages cannot bind to Th1. So there will be no Th1 population, right? And if there's no Th1 population, that means you can't secrete interferon gamma. And if you can't secrete interferon gamma, if you can't secrete interferon gamma, you can't call more macrophages to attack. So what we have to understand here is IL-12 levels will be up, IL-12 receptor is down, interferon gamma is down, Th1 population is down. So the only thing that's up is IL-12. Because IL-12 is secreted by the macrophages normally, right? And here in this IL-12 receptor deficiency, the problem is the receptor is not receptoring. It is not binding. So this IL-12 receptor is on the natural killer cells and on the, for our understanding, Th1 cells. That's how I can remember. You can't know everything. So the IL-12 um, it's on the natural killer cells, and IL-12 receptor is on the natural killer cells, and it's on the Th1 T cell. It's on the Th1 T cell and the natural killer cells. So when IL-12 goes, when IL-12 from the macrophages goes to the Th1 population to make more Th1, it can't. It can't bind because the IL-12 cannot bind to the defective IL-12 receptor located on the Th1 T cell. So this means that there's a decreased Th1 population, and then because there's a decreased Th1 population, that means there is a decreased interferon gamma. If there's decreased interferon gamma, there is a decreased macrophages. So you give, like as doctors, you give interferon gamma. You give interferon gamma because that's what's deficient, that's low. Um, and then because there's interferon gamma, it would respond to the IL-12 and produce more macrophages. So remember I just said that interferon gamma, giving interferon gamma increases the macrophages. Technically, it doesn't increase the macrophages; it activates the macrophages. So I don't know how. I just think I like to simplify things, and I'm like, okay, it makes the macrophages work finally. So I'm like, oh, increase in macrophages. But actually, the number of macrophages do not increase. It activates the existing macrophages. So I don't want you to get an answer wrong because I said it. But so yeah, it doesn't increase. Like if there's like 10 macrophages um, in somebody with interleukin 12 receptor deficiency, and then you give interferon gamma, it doesn't make him 20. It doesn't make the macrophages 20. It makes the existing 10 macrophages more active. That's it. Okay, now we're going to move on to the next section. But before that, verse of the day. Instead, you ought to say, if it is the Lord's will, we will live and do this or that. James 4:15. Now we're going to move on to the antibody structure. Okay, so antibody structure and function. Fab, containing the variable and hypervariable regions, consisting of light and heavy chains. Consisting of light and heavy chains, recognizes antigens. FC port and FC region of IgM and IgG fixes complement. Heavy chain contributes to FC and Fab regions. Right. And light chain contributes only to the Fab region. What does this mean? So Fab, there's two parts to an immunoglobulin. The first part is Fab, which is right here. See this? Fab. This is Fab region, consisting of light chain. See this light chain and heavy chain. So Fab region is this top part that looks like two arms extending out. Actually, it looks like four arms, but think of just what everyone knows what IG looks like. So think of it as two arms reaching out, and that's the Fab region. So I think of it as a fabulous. It's a fabulous part. Because when someone says something is fabulous, they usually are happy, and they dance, they throw their hands up in the air. So I think of it that way. Fabulous. Two arms pointing up, and that is the Fab region of the immunoglobulin. So the light chain is underneath, and the heavy chain is on top. Okay. FC region is the bottom, like the butt of the immunoglobulin. And that area fixes complement. See this FC region right here? So the heavy chain, which is the arm on top, contributes to the FC and Fab region. So heavy chain extends down to the FC region. This heavy chain extends down to the FC region and also makes up the Fab region. Light chain is only part of the Fab region. Okay? So you have to remember the structure of immunoglobulin. I think of it as Fab region. You throw you throw your hands up in the air. So that's the Fab region. It's made up by the heavy chain on top and light chain at the bottom. And that is the fabulous part of the immunoglobulin. The FC region is the butt of the immunoglobulin. And that part is made
Of that part, the FC region is only made up of the heavy chain. Well, that makes sense, right? If something FC region, if that's the immunoglobulin, think of like somebody, a lot of people carry their weight in their butts and thighs. At least that's a desire, right? With all these BBLs going around. So think of it as like heavy chain. Like they're heavy. They're bottom heavy. So I just remembered FC as the end of the immunoglobulin, like the butt, because Fab B comes before C, so B would be like the Fab region would be the lighter part, and C would be like C is after B, and so C would be like the butt region. So from top to bottom, like that. So that's how I think about it. Heavy chain makes up the FC part of the immunoglobulin, and it's heavy. Okay, next. Fab. Fragment antigen binding determines idiotype. Unique antigen binding pocket. Only one antigenic specificity expressed per B cell. Okay. FC. Constant carboxy terminal complement binding carbohydrate side chains. Confers isotype IgM, IgD. Generation of antibody diversity. Antigen independent. Uh, Random recombination of VJ light chain or VDJ heavy chain genes. Random addition of nucleotides to DNA during recombination by terminal deoxynucleotidal transferase. Number 3 would be random combination of heavy chains with light chains. And then generation of antibody specificity, antigen dependent, somatic hypermutation and affinity maturation, variable region. And then isotype switching, constant region. Okay, we'll get into this. So here we said FC region of the IgM and IgE fixes complement. What does that mean?
So, um, antigen on the bacteria binds to the Fab region of IgG, and then Fc region of the IgG activates the complement proteins, which help destroy the bacteria. So if you look here, Fab, that's where the antigen binds. You see this? Antigen binding site. And that's antigen binds to the Fab. So generation of antibody diversity and the generation of antibody specificity are two different things. Um, the antibody diversity is is like when the immune cells they make millions of different antibodies. So they make IgG, they make IgM, um, IgD, IgE, IgA, right? So yeah, that's what that is. And then antigen specificity is when one antibody binds one specific antigen. So that's when the body makes a bunch of B cells that only make one antibody, one type of antibody. Like all these B cells only make IgG, right? And that IgG only binds Staph aureus. So, yes. And then antibody diversity, antibody specificity, both use VDJ recombination. Um, what is VDJ recombination? That's like how the body keeps its immunity up. In the sense, that's how the body makes a lot of different antibodies. So it's like mixing and matching parts of immunoglobulins to make millions of different antibodies. The more antibodies you have, the better it is. Um, so you can fight different types of antigens. Okay. So so VDJ recombination is done to make a maximum diversity of B cells. So there's antibodies against every antigen. And every B cell in the body has a different VDJ combination. So remember we said the VDJ combination is used by both the antibody diversity and the antibody specificity. They're both used by, they both use the VDJ recombination, it's just that they use it differently. Um, the antibody diversity, that one, that one is where the immune system makes millions of different antibodies, each with a unique variable region that can bind to a different antigen. So this is like what we said, different types of antibodies. Antibodies like IgG, IgA, IgM, IgD, IgE. That's all done by the antibody diversity. And that is antigen independent. So antigen diversity, antibody diversity is antigen independent. It doesn't need an antigen. And antibody specificity is antigen dependent. That needs an antigen. Okay. So we said that antibody diversity is when the immune system makes millions of different antibodies, each with a unique variable region that can bind to a different antigen. And antibody specificity is when one antibody binds one specific antigen. So an IgG only binds Staph aureus, as an example. And this it won't bind Clostridium. So this IgG made by the antibody specificity will not bind Clostridium. It can only bind the Staph aureus. Okay. Alright. So under antibody specificity, you have somatic hypermutation and affinity maturation. Somatic hypermutation is where B cells intentionally mutate their antibody genes to improve how well their antibodies bind to an antigen. So somatic hypermutation is where the B cells cut themselves uh, to be able to bind better to the bacteria. Somatic hypermutation only happens in B cells. Somatic hypermutation is a part of affinity maturation. What is affinity maturation? Affinity maturation happens in the lymph nodes is to make better B cells to fight the antigen and be able to bind better. So affinity maturation uh, makes better B cells that are capable of fighting bacteria and can bind well to the bacteria. Somatic hibernation is a process where the B cells cut themselves to be able to bind better to the bacteria. Like a lock and key. If the key doesn't fit, you have to refine the key. So that would be like somatic hypermutation, like fixing the key, cutting off the key, sharpening the key. That's somatic hypermutation. So you can fit the key uh, into the lock. So here the lock would be the B cell that keeps cutting itself to fit better into the lock, which is the bacteria. So somatic hypermutation is a part of affinity maturation. And affinity maturation is what makes produces better B cells to fight the bacteria. So affinity maturation is what makes B cells better. Remember, B cells are made in the bone marrow. Affinity maturation makes those B cells better to fight and bind the bacteria that comes into your body. So first, what happens is B cells cut themselves to bind the bacteria better, and this is somatic hypermutation. And then, in the germinal centers of the lymph node, B cells with better binding survive, and B cells with poor binding die off. This is known as affinity maturation. So somatic hypermutation happens first, then affinity maturation. Then we have the isotype switching, which only happens in the constant region. Isotype switching is when you switch from IgM to any other immunoglobulin, and this only happens in that constant region of the immunoglobulin. So know this. Um, Isotype switching only happens in the constant region. This is the constant region. This blue part right here is the constant region, and that is where isotype switching happens. And somatic hypermutation and affinity maturation only happens in that variable region. So the variable region is right here, here, here. Okay, so remember that. So only the hypervariable region uh, binds the antigen. There is no other part of an antibody that binds the antigen. So only the hypervariable part binds. And I'll show you that again. So right here, this orange. Okay. See it says V, 1, 2, 3, 4. So these are the parts that are the variable regions, hypervariable regions. Okay, so that is the antigen binding site, and that is exactly where the somatic hypermutation and affinity maturation take place. Okay, I know that was a lot, but you'd rather know more information than less, right? Okay. Now, neutralization, opsonization, and complement activation. So this right here, that's a bacteria. This green thing, that's a bacteria. And all these triangles are antibodies that covered the bacteria. So, and it says antibody prevents bacterial adherence. So antibody stops the, there's all those antibodies, 1, 2, 3, 4, 5, 6, 7. There's 7 antibodies on it, on the bacteria. Those antibodies stop the bacteria from attaching itself onto what looks like the intestines. Okay, so here the first step you have the IgG covering the bacteria. Then you have opsonization. Um, this is when bacteria is covered by the IgG. Then the macrophages bind to the Fc region of IgG, so macrophages can eat the bacteria. You see it says macrophage binding up here. This is the Fc portion of the immunoglobulin. See yeah, macrophage binding. Okay. So basically, the butt of the immunoglobulin binds the macrophage, and then this, when the macrophage encounters anything, it eats it. And this right here is the macrophage eating the IgG down bacteria. So the IgG tags it, like it just tags it and be like, oh hey, here's the invader, and gives it to the macrophage, right? So that's what's happening here. IgG coats the bacteria, then calls the macrophages in, and macrophages eat the bacteria. Next, you have complement activation. This right here is antibody activates the complement, enhancing opsonization and lysis. So here you have IgG activating complement, enhancing opsonization and lysis. So So here, antibody activates complement, enhancing opsonization of resistance. So the IgG activates complement proteins, which destroy the bacteria. So you just have to know that IgG activates complement, and complement is a bunch of proteins that come in to destroy the bacteria. That's it. Immunoglobulin isotypes. All isotypes can exist as monomers. Mature naive B cells prior to activation express IgM and IgD on their surfaces. They may differentiate in germinal centers of lymph nodes by isotype switching, which is gene rearrangement induced by cytokines and CD40 ligand into plasma cells that secrete IgA, IgE, and IgE. Affinity refers to individual antibody-antigen interaction, while avidity describes the cumulative binding strength of all antibody-antigen interactions in a multivalent molecule. Okay. So, naive mature B cells are one thing. Naive mature B cells are cells that finish development but didn't encounter an antigen. So they're ready, like they finished maturing, but they have not encountered an antigen. Um, Basically, they're fresh. They developed, but never been infected or got a chance to get infected. One sec. Okay. And these mature naive B cells, these mature naive B cells express both IgM and IgD on their surfaces. The mature naive B cells may differentiate in the germinal centers of the lymph node by isotype switching. Remember, germinal centers are in the cortex of the lymph node, right? Okay. Because that's where the B cells make the antibodies in the cortex of the lymph node, specifically in the germinal centers. So, yeah, germinal centers are where the B cells make the antibodies. So the mature naive B cells differentiate in the germinal centers of the lymph node by isotype switching. Remember, we said isotype switching is when the IgM switches to any other antibody, and this is induced by cytokines and CD40 ligand. So the CD40 ligand is on the CD4 T cell, and CD40 receptor is on the B cell, and this is the second signal for isotype switching. The first signal, remember, for isotype switching is MHC class 2, that's on the B cell, presenting antigen to the CD4 T cell. That's the first signal. Okay, so the first signal for isotype switching is MHC class 2 that is on the B cell presenting antigen to the CD4 T cell. the second signal after this would be CD40 ligand, which is on the CD4 T cell, interacting with the CD40 receptor, which is on the B cell, and then this is the second signal for isotype switching. Then after this happens, the B cells are activated, and then then they undergo somatic hypermutation, isotype switching. They differentiate into plasma cells, and once the B cells differentiate into plasma cells, the plasma cells start secreting IgA, IgE, and IgG. So that's the order of that. Now, the exam, the step one likes to ask very detailed questions regarding immuno. Um, you know, so it's good to know the details, because if you know one detail, you can answer 10 questions that ask the same thing different ways. So, all right. So now once the B cells have differentiated into the plasma cells, the plasma cells will secrete the immunoglobulins, and that is what you see here: IgA, IgG, IgM, IgD, and IgE. Let's start with IgG. IgG is the main antibody in secondary response to an antigen. Most abundant uh, isotype in the serum, fixes complement, opsonizes bacteria, neutralizes bacterial toxins and viruses. Only isotype that crosses the placenta provides infants with passive immunity that starts to wane after birth. IgG greets the growing fetus. Okay, so basically, IgG is the main antibody in the secondary response to an antigen. So what that means is neutralizing means IgG comes and binds to the bacteria. Remember, the IgG coats the bacteria. See this? Neutralization, right? That's all IgG in this. So IgG coats the bacteria. The green thing is the bacteria. Those triangles are IgGs, and they coat the bacteria, rendering the bacteria useless. So the bacteria is floating around the body. IgG goes, grabs them, um, coats them, and then this makes them useless, the bacteria useless. So the first thing that happens is IgG does neutralization, which is coating the bacteria, so bacteria can bind to the body's cells. And the second step is opsonization, where macrophages bind to the Fc region of the IgG, and then macrophages eat the bacteria. The last step is the last step of IgG killing is complement activation. Complement only binds to the Fc part of the IgG. Now you have to realize antigen only binds to the Fab region of the IgG. So remember we said complement only binds to the Fc part of the IgG, which is the butt of the IgG, and antigen only binds to the Fab region, which is like the top of the IgG with the hand stretched out. So you have to know the antigen or the bacteria never binds the Fc portion, never. Complement only binds to the Fc portion, and then macrophages only bind to the Fc portion of the IgG. And now once the complement is bound to the Fc portion of the IgG, the complement starts to kill by activating MAC. We'll get into that. So now, so the complement actually does not travel. It does not travel up to the antigen from the FC part, but I like to think of it one first the antigen binds to the Fab region, then a complement comes and binds to the FC region, and then a complement goes up to kill the to kill the antigen by MAC complex. That's how I think of it. But actually, complement doesn't travel up. The antigen actually uh, it works downstream. It travels down to the complement. But it doesn't matter. You don't have to know which way anything travels. You just have to know that complement goes and kills the bacteria. If you think of it in order of steps, it makes more sense to say that complement goes up to kill the bacteria. Complement goes up to the bacteria to the Fab region to kill the bacteria. Okay, so that's that. That's what fixes complement means. We already did opsonizing bacteria or neutralizes bacterial toxins and viruses. Now, IgG is the only isotype that crosses the placenta. So IgG is the only antibody that crosses the placenta to the baby. So it provides the baby with passive immunity that starts to decline after birth. About 3 to 6 months after the baby is born, the IgG goes away. So the mom gives the IgG to the baby while the baby's in utero, and then that IgG is good even after the baby is born for about 3 to 6 months, and then the baby will start producing his or her own IgG. The mom's IgG goes away after 3 to 6 months after the baby is born. I think of that I think of IgG as the only one that can cross the placenta as Goo Goo Gaga, so it's easier to remember IgG is the only antibody that crosses the placenta to the baby. Now IgA. So IgA prevents attachment of the bacteria and viruses to mucous membranes. IgA does not fix complement. IgA does not fix complement. IgA is a monomer in the circulation or a dimer with J chain when secreted. Crosses epithelial cells by transcytosis, produced in the GI tract, like in the Peyer's patches, and protects against gut infections, like Giardia. Most produced antibody overall, but has lower serum concentrations. Released into the secretions, tears, saliva, mucus, and breast milk. Get released into secretions, tears, saliva, mucus, and breast milk. Picks up secretory component from epithelial cells, which protects the Fc portion from luminal proteases. Okay, so IgA is found in secretions. IgA does not secrete anything. IgA itself is found in our body secretions. IgA prevents attachment of bacteria and virus to mucous membranes. So IgA is a monomer in the blood and a dimer with a J chain when secreted. So a dimer is two monomers put together. Okay. IgA crosses epithelial cells by transcytosis. IgA is produced in the GI tract, um, like in the Peyer's patches, which is in the ileum, and IgA protects against gut infections like Giardia. IgA is the most produced antibody um, in the body, but has lower serum concentrations. IgA is released into secretions like tears, saliva, mucus, and breast milk. So it is the most IgA is the most produced, but um, it's lower in the blood because IgA is released into tears, saliva, mucus, and breast milk. So obviously there will be less concentration in the blood because all of it's going, well, most of it is going into the tears, saliva, and mucus, and the breast milk. IgA picks up secretory component from epithelial cells, which protects the Fc portion from luminal proteases. So um, the IgA picks up secretory component from the epithelial cells, which protects the Fc portion from the luminal proteases. The proteases are something that it breaks down proteins. So what they're saying is mm, the Fc portion of the IgG, which is the butt of the IgG, is hidden because the secretory component covers that area. So the secretory component in the epithelial cells covers the Fc portion of IgG because IgG does not want um, the Fc portion to be broken down from does not want the Fc portion to be broken down by proteins, by proteases, is because that's what breaks down proteins, right? Because immunoglobulin, like IgA, is a protein, um, so that is a target for proteases. Okay, so IgA is going to protect its Fc portion from the proteases. And you have to know IgA protects the body by removing bacteria from the gut. And you have to know J chain. Um, the J chain. So IgA has a J chain when the IgA is secreted by the mucous membranes, right? So IgA is a dimer when secreted by mucous membranes, and this um, and this IgA when it's secreted by the mucous membranes, it's a dimer with J chain. Okay, you should know that. And a J chain is something that joins two immunoglobulins together, as you see here. See this J chain. This is what it looks like when the IgA is being secreted into breast milk. Okay, all right. Next, IgM. IgM is produced in the primary response to an antigen. IgM fixes complement. There is an antigen receptor on the surface of I mean, it is an antigen receptor on the surface of the B cells. IgM is a monomer on the B cell, pentamer with J chain when secreted. The pentamer enables avid binding to antigen while humoral response evolves. Okay. Okay, so IgM is produced in the primary response to an antigen, which means uh, IgM is the first antibody that's immediately produced in response to an antigen. IgM fixes complement. IgM is on the B cell and allows the B cell to recognize and bind antigen. So IgM serves as the antigen receptor on the surface of B cells. IgM is a monomer on the B cell, and IgM is a pentamer with the J chain when secreted, which means right here, this is the design that IgM looks like when IgM is secreted, and this J chain right here, this red loop is a J chain. Okay, so it has a J chain when it is secreted. So the J chain is what joins all 5 parts to form the pentamer. All 5 IgMs are together to form a pentamer with the J chain when IgM is secreted. And pentamer enables avid binding to antigen while humoral response evolves. Pentamer formation of IgM allows tight binding to antigen while loading a Th2 response. So basically, they're saying pentamer, the pentamer formation of IgM allows a tight binding to antigen while loading a Th2 response. So having this 5 IgM compound with the J chain allows the IgM to bind the antigen better while the body activates a Th2 response. So a Th2 response is when the body activates B cells to make antibodies, right? Th2 Th2 response comes from CD4 T cells, and that is humoral response. And Th1 also comes from CD4 T cells, and that is a cell-mediated response. But here, in the context of IgM, we are only talking about humoral response, which is Th2 response, which means the B cells make the antibodies, right? Okay, so that's what that is. IgD, unclear function, found on surface of many B cells and in serum. Okay, so no one really knows what IgD does, but IgD is found on the top of many B cells and in the blood. It, you know, it is randomly there with, I believe it's IgM. So IgM and IgD are together on B cells. Okay, IgE binds mast cells and basophils, cross-links when exposed to allergen, mediating immediate type 1 hypersensitivity, the release of inflammatory mediators like histamine, contributes to immunity to parasites by activating eosinophils. So IgE binds mast cells and basophils, cross-links when exposed to allergen, mediating immediate hypersensitivity, like mediating immediate type 1 hypersensitivity through release of inflammatory mediators such as histamine. Contributes to immunity to parasites by activating eosinophils. Okay, so this is what happens here. Uh, you see, this this right here is this is a mast cell. Okay, the IgE is secreted by the plasma cell in response to an allergy the patient is having. Then, once these IgEs are secreted by the plasma cell, the IgE goes and binds to the mast cell. And then, if enough IgEs bind, that's known as like cross-linking of the IgEs. That will trigger histamine release. So I'll walk you through the steps. Plasma cells secrete IgE in response to an allergen or parasite. IgE binds to mast cells and the basophils with IgE's Fc region before any allergen is even in contact. Later, when the same allergen enters the body, that allergen binds to the Fab region of the IgE that's already sitting on top of the mast cell. And then, if enough IgEs uh, start gathering on top of start binding on top of the mast cells, that's known as cross-linking of IgEs. Um, and once enough IgEs cross-link, this will trigger mast cell degranulation. And this is what releases the histamines from the mast cells, causing anaphylaxis. Okay, so that's what this means. IgE binds mast cells and basophils, cross-links when exposed to an allergen, mediating immediate type 1 hypersensitivity through the release of mediators such as histamine, contributes to immunity to parasites by activating the eosinophils. So what that means, contributes to immunity to parasites by activating eosinophils. IgE binds parasites, and that causes a degranulation. So if somebody has asthma and parasites, their IgE will be up. So what happens here, specifically, where it says contributes to immunity to parasites by activating eosinophils, this means the IgE binds parasites. This leads to degranulation. How? So the Fab region of the IgE binds to antigens on the parasite, and then the Fc region of the IgE binds to the eosinophil. When multiple IgEs are bound to the parasite, cross-linking activates the eosinophils. So cross-linking of IgE activates the eosinophils. Eosinophils release major basic protein to kill the parasites. So the Fab region of IgE, which is the antigen grabbing area of the IgE, that Fab region grabs the antigen on the parasite, and then the butt of the IgE, which is the Fc region of the IgE, binds to eosinophils. So the goal of the IgE is for the antigen is the goal of the IgE is for the IgE to grab the parasite and attach it to the eosinophil when it comes to parasites. Yeah, so the IgE grabs the parasite and attaches it to the eosinophil. That's the IgE is the mediator here. So the IgE grabs the antigen of the parasite with its Fab region and attaches itself to the eosinophil with the IgE's Fc region. And when this happens, there is cross-linking of IgEs on top of the eosinophil, and then the eosinophils are activated when this releases major basic protein, and this kills off the parasite. Okay, so that's what this is. That's when it comes to parasites. And then what we discussed earlier is when it comes to allergies. Yeah, allergies. So it's two different. It's the same mechanism. It's two different ways. So when it comes to anaphylaxis, it's allergy. Mast. We have mast cell degranulation, which releases histamine, and that causes anaphylaxis. And when it comes to parasitic infections, um, eosinophils are the ones that are bound by the IgE, and the eosinophils release major basic protein that kills off the parasite. So in asthma and parasites, IgE will be up because IgE is what cross-links for both of the scenarios on top of whether it's the mast cell or whether it's the eosinophil. Okay, all right, bye. So thymus independent antigens are antigens um, lacking a peptide component. For example, lipopolysaccharides from gram-negative bacteria. Thymus-independent antigens cannot be presented by MHC to T cells. They are weakly immunogenic. Vaccines often require boosters and adjuvants. For example, capsular polysaccharide subunit, Streptococcus pneumonia PPSV23 vaccine. So thymus-independent antigens, they're saying that they don't need the thymus. What's in the thymus? T cells. So antigens lacking a peptide component. So these are antigens lacking a peptide component, means that there is no protein there, right? They don't. These antigens don't have a protein part, and they can only make IgM antibody. So we said thymus independent antigens doesn't need T cells, and it can only make IgM antibody. So T cell can only recognize an antigen that's on a protein like MHC1 or 2, and only IgM can be made here in the thymus independent antigen part. So what they're saying is, T cells need T cells need an antigen that's on a protein, so that T cells can work. And since there is no protein here in the thymus independent antigen, T cells cannot work here. So, so vaccines often require boosters and adjuvants like capsular polysaccharide subunit of Streptococcus pneumoniae (PPS) V23 vaccine. This PPS V23 vaccine, it does not work on T cells, so it has a short immunity because no T cells are present. What they're saying is like you vaccines need boosters and adjuvants. Right? Remember how we get a vaccine, then you had to get a booster. Um, they need T cells to have longer immunity, but here in thymus independent antigen, they do not have the T cells, so they have short immunity. And an example of that short immunity type of vaccine would be PPS V23, because PPS V23 does not have the T cells. And thymus dependent antigen, antigens containing a protein component like in Streptococcus pneumonia PC V13 vaccine, polysaccharide polysaccharides conjugated to diphtheria toxin like protein. Class switching and immunologic memory occur as a result of direct B cells with Th cells. So thymus dependent antigen means you need T cells. Yeah, you need T cells, and T cells can only recognize the antigen if the antigen is a protein structure. So remember we said thymus depends. So this one depends on the thymus. What's in the thymus? T cells. Uh, you need T cells for the thymus dependent antigens. And remember how we said T cells can only recognize antigen if the antigen is on a protein. So the Streptococcus pneumonia PCV13 vaccine is a conjugated vaccine that has the T cells. So this vaccine has longer immunity. The polysaccharides conjugated to diphtheria toxin-like protein. This whole thing is a protein, okay? If antigen is only made of polysaccharides in a vaccine, then only IgM will be produced because polysaccharides are only sugars. And remember we said to have a humoral response with IgG, IgM, IgA, IgE, you need T cells. You need the T cells to interact with the B cells. Remember, we talked about this. Th2 interacts with Th2, which is a CD4 cell, interacts with B cells. Yeah, yeah. Th2 is a CD4 T cell that interacts with the APC, which would be the B cell. So remember how we said CD4 needs two signals to be activated to produce G, IgM, IgE, IgA. The first signal will be MHC2. MHC2 binds T cell receptor on the CD4 T cell. So the first signal is MHC2 on the B cell will bind T cell receptor on the CD4 T cell. The second signal for the B cells to be activated is B7 on the B7 on the B cell binds to CD28 on the CD4 T cell. Now the CD4 T cell is fully activated. So, like we said, um, remember we said CD4 needs two signals. We just talked about it. The MHC2 on the APC binds the T cell receptor on the CD4 T cell, and the second signal is B7 on the APC binds to CD28 on the CD4 positive T cell. Now the CD4 positive T cell is fully activated. And now the CD4 positive T cell can interact with the B cell for class switching. Uh, now, now we have an activated CD4 positive T cell, and we have another B cell. So the B cell swallows the antigen to present on MHC class 2 to the CD4 T cell, and then now the CD4 T cell recognizes the antigen on the MHC class 2 on the B cell, and then you have the CD40 receptor on the B cell that binds to the CD40 ligand on the CD4 positive T cell. And once this happens, the CD4 positive T cell secretes the cytokines um, and determines immunoglobulin class switching of the B cells. The B cells are activated, undergo class switching, and producing IgG, IgM, IgA, like that. So this is like a whole process, and it took me a long time to understand. But if you understand this, you understand immunology. So CD4 positive T cell needs two signals. The first signal is what? MD MHC2 on the APC binds with T cell receptor on the CD4 positive T cell, and the second signal is B7 on the APC binds to CD28 on the CD4 positive T cell. And now CD4 positive T cell is fully activated. Now the next step to get to to make the antibodies is this. once the CD4 positive T cell is fully activated, um, it tries to talk to the B cells, the B cell. Um, the B cell has MHC2 class on it, which has the antigen on it. So this activated CD4 positive T cell um, recognizes this MHC class 2 by using the T cell receptor on the CD4 T cell. The next step is CD40 receptor on the B cell binding to CD40 ligand on the CD4 positive T cell. And now you get immunoglobulins. Okay, so that entire step is immunoglobulin. Um, it's how immunoglobulins are produced. Okay, so here, what they're saying is thymus-dependent antigens, class switching, and immunologic memory occur as a result of direct B cells with Th cells. Th cells are CD4 positive T cells. So what they're saying is class switching. Like we just said, that's producing how the body produces immunoglobulin and makes memory cells. That happens as a result of direct contact of the B cells with CD4 positive T cells, like we just said. So for the B cells and the CD4 positive T cells to interact, uh, you first need the T and B cell activation, which we said was CD4 having two signals, and then the next step would be the B cell activation and class switching. So if you don't have T cells, you don't then you don't have CD4 T cells, and then you won't have anything to interact with the B cells to make immunoglobulins. So that's why they're saying thymus-dependent antigens. So this thymus-dependent antigens depend on the T cells. So Streptococcus pneumonia PCV13 vaccine depends on T cells, and so does polysaccharide conjugated to diphtheria toxin-like protein. Both of these vaccines depend on T cells to make immunoglobulins. That's all they're saying. Is it a simple concept? Probably. Not in my mind though. But that is what they're saying. So, um, like we said here, the polysaccharides conjugated to diphtheria toxin-like protein. Polysaccharides conjugated to diphtheria toxin-like protein means polysaccharides are added to the diphtheria toxin-like protein. And why are they added? Okay, if antigen is only made of polysaccharide in a vaccine, then only IgM will be produced because polysaccharide is just sugar. There's no protein. And so if it's only polysaccharides, the T cells won't be activated. But if we add a protein to these polysaccharides, which is what we did here, polysaccharides conjugated to diphtheria toxin-like protein, the vaccine, uh, this vaccine, now has T cells in it. And then since it has T cells, you're able to do class switching, and you can make memory T cells that can be up, which is what we want. That's how you increase immunity, right? Yes, that's it. So, complement is a system of hepatically synthesized plasma proteins that play a role in innate immunity and inflammation. Membrane attack complex MAC defends against gram-negative bacteria. The CH50 test is used to screen for activation of the classical complement pathway, activation pathway, classic IgG or IgM mediated, alternative pathway, microbe surface molecules, lectin pathway, mannose or other sugars on microbe surface. Functions C3b does opsonization, C3a, C4a, C5a does anaphylaxis, C5a does neutrophil chemotaxis, C5b to 9 does cytolysis. Opsonins C3b and IgG are the two primary opsonins in bacterial defense, enhance phagocytosis. C3b also helps clear immune complexes. Inhibitors decay accelerating factor DAF, AK, CD55, and C1 esterase inhibitor help prevent complement activation on self cells, like red blood cells. So IgG binds to pathogen. Fc portion of IgG binds to the macrophage, and complement activation leads to C3b being deposited on the pathogen surface, which also helps the macrophage recognize and eat the pathogen. So IgG is binds to the bacteria, and then the Fc portion of IgG is what binds to the macrophage. And once that happens, uh, the complement like C3b begins to deposit on the bacteria's surface, and this tells the macrophage to eat the bacteria. Okay. Mm. So remember, neutralization is the covering. Um, neutralization is uh, covering the bacteria to block and prevent the bacteria from binding to the cells. Opsonization is marking the bacteria with molecules like IgG or C3b so that phagocytes can grab and destroy the bacteria. Okay, all right. So over here, yeah, we said C3b is what opsonizes. So C3b marks the bacteria with IgG and C3b so that macrophage can come and eat the bacteria. C3a and C4a and C5a would be anaphylaxis. What causes the anaphylaxis. C5a is what calls the neutrophils in. C5b to 9 would be MAC. That's what causes cytolysis, also known as MAC complex. So the membrane attack complex that will poke holes on the cell membrane of the bacteria. Okay. Okay, so and this is the pathway here of the complement pathway. Um, Here we see the MAC complex do this. Here we have C5b to 9. And then we have the lectin pathway. Lectin will come bind to mannose. This will cause a C1-like complex, and it will follow the same steps as classic pathway. So lectin will come bind to mannose. This will cause a C1-like complex, and this will follow the same steps as classic pathway. Alternative pathway is spontaneous and microbial surfaces, um, amplifies generation of C3b. Classic pathway does antigen antibody complexes. So the goal um, of the pathways, we're going to talk about that. All three pathways lead to C3 convertase formation, which cleaves C3 into C3A and C3B, kicking off the complement cascade that ends in opsonization, which is done by C3B. It also increases the C3A and C5A, and also um, results in MAC complex formation, which is C5B to C9. And so each of these pathways, we're going to start with the alternative pathway. Here, if C3b binds to a bacteria surface, it activates complement. Alternative pathway is always on. It's part of the innate immunity, so no antibodies are needed. It's always there since birth. Alternative pathway. So here C3b binds to a bacteria and activates the complement. So remember how we said complement deposits it directly on the bacteria, right? It covers the bacteria, right? That's what's happening here. And then once the complement is covered, complement covers the bacteria, um, it activates the rest of the pathway, resulting in MAC formation at the end. Now, lectin pathway. Bacteria rich in mannose uses mannose binding lectin and activates C4 and C2. No antibody needed here either. Here, mannose isn't found in a lot of bacteria, so that's what this pathway uses, lectin. So bacteria rich in mannose uses mannose binding lectin and activates C4 and C2. No antibody needed. And in classic pathway, this is where you see the antibodies, right? IgG and IgM are bound to bacteria, and then C1 binds to Fc region of IgG. So IgG and IgM are bound to the bacteria, and then C1 binds to the Fc region of IgG. Right. So remember how we said antibodies come and cover the bacteria. This is a part of neutralization, right? And then C1b binds to the Fc region of the antibody, and then the macrophage knows to bind to the um, FC region as well. So the IgM binds to the bacteria at the Fab region, which is the top part of the antibody, and then C1 binds to the Fc region of IgG. Then this entire pathway happens to this classic pathway, then it results in MAC.
So early complement deficiencies, C1 to C4, there is increased risk of severe recurrent pyogenic sinus and respiratory tract infections, increased risk of SLE. So So you have a deficiency of C1 to C4. You cannot clear encapsulated organisms, especially pneumonia. That's why you have recurrent pyogenic sinus and respiratory tract infections. There's increased risk of SLE. SLE is uh, because of many immunoglobulins in the blood, because there are no C1 to C4 to clear the immunoglobulins, so they build up. So the reason people have SLE with C1 to C4 deficiency is because there's a lot of Ig in the blood, and there is no C1 to C4 to clear all that Ig, so all the immunoglobulins build up, right? SLE is when the body attacks itself, the antibodies.
So early complement deficiencies, C1 to C4, increased risk of severe recurrent pyogenic sinus and respiratory tract infections, increased risk of SLE. Okay, so here, early complement deficiency, C1 to C4 deficiency, you have antigen antibody complexes that build up. The problem is the body cannot clear these antigen antibody complexes. So when there's a deficiency of C1 to C4, there will be decreased complement activation, and this causes immune complexes to build up and deposit in the tissues. There will be decreased phagocytosis, so there will be increased immune complexes circulating in the body, causing SLE. I'll repeat it. When there's a deficiency of C1 to C4, there will be decreased complement activation, and this causes immune complexes to build up and deposit in the tissues. Then there will be decreased phagocytosis, so there will be increased immune complexes circulating, causing the SLE. Right, because if there's your complement cannot be activated, so um, the body can't clear these extras that come into the body. Um, and since there's no complement, there is nothing, there's no complement to bind to the Fc portion of the immunoglobulin, and and then the macrophage can't come and attach because macrophage doesn't know to attach because there's no complement. So uh, the macrophage won't be able to phagocytize the bacteria or the immune complexes, right? And this will cause pneumonia, especially, um, you know, the body can clear encapsulated organisms that cause pneumonia, and the antigen antibody complexes build up, causing lupus. Next, we have terminal complement deficiency. This is C5 to C9 deficiency. You have increased susceptibility to recurrent Neisseria bacteremia. So C5 to C9, this forms the MAC, and MAC lyses pathogens. And if MAC is deficient, there will be decreased ability to kill the bacteria, especially Neisseria. So C5 to C9 forms the MAC complex. If you don't have C5 to C9, you cannot form the MAC complex. So you cannot lyse the bacteria. So the bacteria will build up, especially gram-negative bacteria like Neisseria. Next, you have complement regulatory protein deficiencies. The first one is C1 esterase inhibitor deficiency. This causes hereditary angioedema due to unregulated activation of kallikrein. This increases bradykinin. This is characterized by decreased C4 levels. ACE inhibitors are contraindicated because they also increase bradykinin. Okay, so C1 esterase stops C1 and kallikrein normally. So C1 esterase stops C1 and kallikrein normally. So if there is a C1 esterase deficiency, kallikrein and C1 activity and the bradykinin will go up. So there will be angioedema, and this is an emergency because this will cause swelling of the lips, throat. Yeah, swelling of the lips and throat. So we have C1 esterase inhibitor deficiency. So when you have a C1 esterase inhibitor deficiency, you'll have a lot of kallikrein build up, and that will increase the bradykinin, and this will cause angioedema. And you cannot give ACE inhibitors in somebody with a C1 esterase inhibitor deficiency because ACE inhibitors also increase bradykinin. So that's like a double whammy, right? So because you already have increased bradykinin from the C1 esterase inhibitor deficiency, and if you also give ACE inhibitor, that will also increase the bradykinin, causing angioedema. So before I said C1 esterase I forgot the inhibitor. So I'll just say it again. Um, I said C1 esterase stopped C1 and kallikrein. That's wrong. It's C1 esterase inhibitor. The inhibitor is what stops, right? So C1 esterase inhibitor stops C1 and kallikrein. So if C1 esterase inhibitor is deficient, the kallikrein, C1 activity, and the bradykinin will go up. So there will be angioedema. I'll say it again. C1 esterase inhibitor deficiency causes hereditary angioedema due to unregulated activation of kallikrein, and this increases bradykinin. So you have to know what C1 esterase inhibitor normally does. C1 esterase inhibitor normally stop C1 and the calcrete so that you don't get angioedema. But if that esterase is deficient, so if C1 esterase inhibitor is deficient, you will get increased calcrete, increased C1 activity, and increased bradykinin, which will cause angioedema, which is an emergency. So bradykinin causes vasodilation, and that increases the vascular permeability, which is what causes the angioedema. You will have decreased C4 levels in C1 esterase inhibitor deficiency as well, because the C4 um, is being okay. The C4 is being eaten up by the increased C1 activity here. So normally C1 normally cleaves C4, but in C1 esterase inhibitor deficiency, because there is so much C1 activity, the C1 activity is so increased it consumes the C4, so it eats the C4. So that's why there is decreased C4 levels in C1 esterase inhibitor deficiency. ACE inhibitors are contraindicated because both ACE inhibitors um, and C1 esterase inhibitor deficiency increase bradykinin, which causes swelling and angioedema. Okay, that makes sense, right? C1 esterase inhibitor is what stops C1 and calcrete from building up. So if that inhibitor is not there, the C1, the calcrete, and the bradykinin will build up, causing angioedema. And because there is so much C1 activity, it's going to eat the C4 levels. So you'll have decreased C4 levels. Next, we have paroxysmal nocturnal hemoglobinuria. This is a defect in the PIGA gene, preventing the formation of glycosyl phosphatidylacetosol GPI anchors for complement inhibitors, such as decay accelerating factor, DAF, and CD55, and membrane inhibitor of reactive lysis, MIRL, and CD59 causes complement-mediated intravascular hemolysis. Okay, let's see. So what this means is PIGA is important for making GPI anchors, right? PIGA is important for making GPI anchors. GPI anchors are special molecules that attach certain protective protein to the surface of the red blood cell, like DAF and MIRL. MERL. If no DAF or MERL, then complement will attack the RBC membrane. If no DAF, C3 convertase will attack the RBC membrane. If no MERL, MAC can't be stopped, and MAC will poke holes in the RBC membrane, and this will cause intravascular hemolysis, which will lead to decreased haptoglobin and dark urine. Patient with early morning hemoglobinuria due to increased complement activation during sleep, because there is increased CO2 and decreased pH during sleep, and this acidosis activates complement system. There is lysis overnight. The red color is due to hemoglobin, not whole RBCs. One RBC has millions of hemoglobin, and each hemoglobin has 4 oxygens on it. Okay, that's just a side note I made. Okay, so what they're saying is DAF and MERL protect the red blood cell from complement activation. So in PIGA defect, our red blood cells are destroyed, So there is hemolysis, which is why you get this dark urine on the side. You see that? That's not red blood cells. There's no red blood cells there. It's busted red blood cells. So it's like hemoglobin. Um, that's there. Um, so when you sleep, there is CO2 retention because the lungs don't work like in daytime. So when you sleep, you have like uh, hypoventilation, and this will cause increased CO2, which is acidic. So that's why overnight this acidosis will um, acidosis increases complement. So when you sleep, if you have increased acidosis, that's going to increase that's going to activate the complement in your body, and this will cause hemolysis. Okay, so um, just to be clear, PAGA is important for making GPA anchors. GPA anchors are special molecules that attach certain protective proteins to the surface of the red blood cell, like DAF and MERL. If there is no DAF or MERL, then the complement will attack the RBC membrane. If no DAF, DC3 convertase will attack the RBC membrane. If no MERL, MAC cannot be stopped, and MAC will poke holes in the RBC membrane, and this will cause intravascular hemolysis. You will see decreased haptoglobin and dark urine. So patient with early morning hemoglobinuria due to increased complement activation during sleep because there is increased carbon dioxide and decreased pH, and this acidosis activates the complement system. There is increased lysis overnight, increased lysis of the RBCs overnight. The red color is due to the hemoglobin, not whole RBCs, right there in that urine sample. One RBC has millions of hemoglobins, and each hemoglobin has 4 oxygens on it. So haptoglobin is a protein made by the liver, and haptoglobin's job is to bind free hemoglobin that's released into the blood when RBCs are destroyed. So haptoglobin is made in the liver, and haptoglobin's job is to bind random hemoglobin that's just floating around in the blood. So here, since so many RBCs are destroyed, there is a lot of hemoglobin that's floating around, and this haptoglobin will go bind this hemoglobin, and that's why there is a decreased haptoglobin. And so there's just one other point before we move on to cytokines. C1 to C4 deficiency, you're more susceptible to get Haemophilus influenza type B and Streptococcus pneumonia. And then in C5 to C9 deficiency, you get Neisseria infection. Remember that. The conjugated vaccines are made for Haemophilus influenza type B, Streptococcus pneumonia, and Neisseria meningitis. Okay, now we're going to go into cytokines. So, important cytokines. Acute would be IL-1, IL-6, TNF alpha, and then recruit IL-8, IL-12. Okay. Interleukin-1 causes fever, acute inflammation, activates endothelium to express adhesion molecules, induces chemokine secretion to recruit WBCs, also called osteoclast activating factor. So IL-1 causes fever and acute inflammation, and interleukin-1 activates the endothelium to express adhesion molecules. Yeah, it activates the endothelium to express adhesion molecules. Interleukin 1 induces chemokine secretion to recruit white blood cells. And interleukin is also called osteoclast activating factor. So, macrophages and T cells secrete IL-1, which increases CAM, that's CAMS, on the endothelium of the blood vessel. By the WBCs attaching with the CAMs, there will be increased WBCs that can come and help with inflammation. WBCs attach to CAMs on the blood vessel wall, and this is overseen by interleukin 1. WBCs attaching to CAM allow the WBCs to enter the tissue to fight the inflammation. CAM, it just means cell adhesion molecules. So interleukin 1 recruits the WBCs. Interleukin 6 causes fever and stimulates production of acute phase proteins. So interleukin 1 and interleukin 6 involved in fever. Tumor necrosis factor alpha, this is TNF alpha. This activates the endothelium, causes WBC recruitment and vascular leaks. So TNF alpha causes the inflammation. It helps with inflammation and helps WBC attach to CAM. So TNF alpha causes inflammation and also helps with inflammation and helps WBC attach to CAM. So it brings the WBC into the tissues for the body to help fight the um, infections. TNF alpha causes vascular leaks due to increased inflammation. Vascular leak causes edema. Right, so if there's inflammation, um, then there will be leaks, right? And these vascular leaks will cause edema. TNF alpha causes cachexia in malignancy, which is a muscle wasting. So TNF alpha maintains granulomas in TB. So this means that TNF alpha keeps the granulomas of TB closed. So TNF alpha keeps the granulomas of TB closed and latent, right? It keeps it away, closed. Um, TNF alpha, IL-1, IL-6 can mediate fever and sepsis. Interleukin 8, it's a major chemotactic factor for neutrophils. Clean up IL-8. Neutrophils are recruited by IL-8 to clear infections. Right. So it attracts the neutrophils to come to the site. The four neutrophil attractors are what? C5a, LTB4, IL-8, and bacterial products. What are they? C5a, LTB4, IL-8, and bacterial products. And remember, C3b is for opsonization. C3a, C4a, and C5a are seen in anaphylaxis. C5a is seen in neutrophil chemotaxis, and C5b to 9 is the MAC complex, which pokes holes in the enemy. Okay, next we have interleukin 12. Interleukin 12 induces differentiation of T cells into Th1 cells, and interleukin 12 activates the natural killer cells. So interleukin 12 increases the Th1 population. What is a Th1 population? That is the split off from CD4. Th1 increases the macrophages in the body, right? Remember the chart that we talked about? CD4 T cells turns into helper cells, and helper cells turn into Th1, Th2, and the third one, which is Th17 and T regulatory cells. Yeah, so Th1 increases it activates the macrophages and the cytotoxic T cells. Okay, so interleukin 12 increases the Th1 population and also activates the natural killer cells. Interleukin 2 stimulates growth of helper T cells, cytotoxic T cells, and regulatory T cells, and natural killer cells. Okay. Interleukin 3 supports growth and differentiation of bone marrow stem cells. So interleukin 3 supports growth and differentiation of bone marrow stem cells. Interleukin 3 functions like granulate macrophage colony stimulating factor. So this granulocyte macrophage colony stimulating factor produces WBCs. So interleukin 3 makes WBCs also, because interleukin 3 uh, makes the bone marrow stem cells, and that would include the WBCs. Different, yeah, okay. So next we have interferon gamma. This is secreted by natural killer cells and T cells in response to antigen or IL-12 from the macrophages. Stimulates macrophages to kill phagocytosed pathogens. Inhibits differentiation of Th2 cells. So interferon gamma is secreted by the natural killer cells and the T cells in response to antigen or IL-12 from the macrophages. And this uh, interferon gamma stimulates the macrophages to kill phagocytosed pathogens. Interferon gamma inhibits the differentiation of Th2 cells. Interferon gamma also activates natural killer cells to kill virus-infected cells. Interferon gamma increases MHC expression and antigen presentation by all cells. interferon gamma activates macrophages to induce granuloma formation. Next, we have Th2 cells. Interleukin 4 induces differentiation of T cells into T helper cells. Th2 cells. So interleukin 4 induces differentiation of T cells into Th2 cells. Interleukin 4 promotes the growth of B cells, and interleukin 4 enhances class switching to IgE and IgG. So interleukin 4 helps with B cell growth by telling B cells to switch from IgM to IgE and IgG. Remember, IgM is the first immunoglobulin, then B cells undergo class switching to become IgE, IgG, IgA. So interleukin helps with class switching from IgM to IgE and IgG. Interleukin 5 promotes growth and differentiation of B cells. Interleukin 5 enhances class switching to IgA, stimulates growth and differentiation of eosinophils. Interleukin 5 promotes the B cell growth by telling B cells to class switch to IgA. So interleukin 5 tells the B cells to class switch from IgM to IgA. Interleukin 10 attenuates inflammatory response. It decreases expression of MHC class 2 and Th1 cytokines. Interleukin 10 inhibits activated macrophages and dendritic cells. Interleukin 10 also secreted by regulatory T cells. So TGF-beta and IL-10 both attenuate the immune response. Okay, so TGF-beta and IL-10 both decrease the immune response. Their job is to make peace. TGF-beta and IL-10 both decrease the immune response. So remember how we said here it says, interleukin 10 inhibits activated macrophages. Remember, the macrophages produce inflammatory response, right? So IL-10 does the opposite of that. It stops the macrophages. So macrophages normally secrete IL-12, and the IL-12 receptor is on the T cell. So the macrophages secrete the IL-12, and the IL-12 receptor is on the T cell. The binding of the IL-12 that the macrophages secrete to the IL-12 receptor that's on the T cell causes increased Th1 cells. And this increased Th1 cells produce interferon gamma, and this activates more macrophages, and this causes intracellular killing, and macrophages make more IL-12, and this is a positive feedback loop. So this is an inflammatory process, right? The macrophages binding. Okay, so the macrophages binding to the T cells through IL-12, and then that interaction causing more Th1 cells to happen, and this Th1 cell making a lot of interferon gamma, which calls more macrophages to the site. And if you call more macrophages, what does that do? It kills. There's more killing going on. And then, what do macrophages secrete? IL-12. And this is a positive feedback loop. So the IL-12 um, is being made by the macrophages, and then when IL-12 is being made, directly it causes the Th1 cells to be made, from the interaction of um, the interaction of the macrophage and the T cell, and then that causes, once more Th1 cells are made, that causes more interferon gamma cells, interferon gamma to be made. And if more interferon gamma is made, that's going to cause more macrophages to come. And if there's more macrophages, it goes back to more IL-12 being made. And this is a positive feedback loop that keeps going, going, going. But IL-10 opposes this process we just talked about. Now, TNF alpha inhibitors used in Crohn's disease, right? If you use the TNF alpha inhibitor, that would cause a reactivation of TB because there's no more TNF alpha to maintain and keep the granuloma closed in latent TB patients. So remember, TNF alpha inhibitors reactivate TB. And now we're moving on to respiratory burst. So this interferon gamma, the Th1 cells secrete the interferon gamma. And once the Th1 cells secrete the interferon gamma, that will call more macrophages on site, and the macrophages will continue killing, and there's more killing, and then all these macrophages that are not called on site will produce more IL-12, and this is a positive feedback loop. I know I've said that a lot of times, but I'm telling you, if you don't say it like 100 times, you will forget. Okay, now we can actually move on to respiratory.